Thank you, Jillian, and thank you to the Academy, Claire Palmer, and Peter, everybody who's done such a fabulous job to bring this together every year now. I think I've been here since 2012, and the Academy has stuck with me through thick and thin, and so it's always a delight to come to London and talk to the patients, and I hope we'll do it for many years in the future. So when we thought about this meeting, we've done a series and in what we call lifting the veil, and that is taking the mystique away from these diseases, the misconceptions about what they are and what they aren't, and this time what we're going to talk about with really just world-renowned speakers, as you'll hear from the rest of the day, is just stepping outside the box. How do we think outside the box in order to treat these diseases quickly, recognize them quickly for what they are, and move forward and save our children, save generations, save our families, because it's really everybody. What I do and have done for essentially all of my adult life is think about immune therapy. I think about the immune system, I think about the pathogens, because that's how we understand normal immune function, is to study disease and to treat disease with what we used to call biological response modifiers. So this Time magazine from the cover of March 31st, 1980, was just before I graduated from college, and my degree was in nutritional biochemistry, and that's where I studied a lot of natural products chemistry and thinking of how to treat cancers with our own immune system and teach our own immune system to function. Well, I was going to take a year to cure cancer with interferon there. That was the magic bullet of cancer, and then go back to medical school. And of course, I never made it back to medical school. I ended up with a PhD because my mentor, Dr. Rossetti, convinced me that you could help a lot more people with drug development than what we could do one-on-one, and that if we worked together with clinicians, we could explain a lot of things. Well, lo and behold, last month, this was the cover of Time magazine, and what they're talking about was a new kind of immune therapy called checkpoint inhibitor therapy, and all you really have to know about that is what we've had to do in order to treat these diseases is simply take the brakes off the immune system and let all hell break loose is basically what happens. They say brutally selective. And what people fail to realize is that immune therapies are drugs, and they interact with the immune system in very different ways, and it's not simply going to be a magic bullet, or it's going to be more personalized medicine. So what have we learned in those 36 years about the immune system? Well, what we're going to be talking about today is the interactions between the pathogens and the frontline defense, which is your innate immune response. And what we've learned over the last 36 years is that there are many pattern recognition receptors and danger molecular patterns on the surface of cells. And the bacteria are recognized not so much specifically but non-specifically based on these pathogen-associated molecular patterns. And so what this intends to show you, we're going to talk today about Borrelia, and that's a class we call gram-negative bacteria. But Borrelia doesn't live alone in your immune system, as far as it's not a vacuum where the immune system only sees Borrelia, and there's a linear signaling of a single pathway, but what it sees is the entire environment, the immune system, and they're not linear at all. They're overlapping and redundant in such ways that they can stimulate what we will call these cytokine storms. So we learned all about these cytokines, and these are just signaling molecules. Once that recognition is made, it triggers the production of these cytokines, and then the cytokine tells the adaptive immune response what to do. What am I looking for? Am I looking to make mucus production, say in a mast cell, or histamine reaction? Am I looking to clear extracellular pathogen? What did that antigen just tell that antigen presenting cell, the dendritic cell, and what else were these two cells doing as they talk together? And it's many more than two cells. This is as simplistic as I can make it, and again, it's not linear at all. So when we first started doing immune therapy with interferon back in 1980, Frank Rossetti, my mentor, had just isolated the first human cancer-causing retrovirus, and in order to do that, he had to isolate a factor that would grow T cells, because at the time the T cells did absolutely nothing, and the B cell and the antibody production was everything. So in 30 years now we know there's a huge plasticity in the immune response, and it's not one thing and one end result. It's a threshold, it's a gradient of signaling that is a tipping point. And so we're going to be thinking a lot about that as we think about what we know about Borrelia, and you'll hear a lot about that later today from Armin and others, how it interacts with the immune system, a lot of which is completely unknown, but how it actually talks to the rest of the immune system, but also the milieu of vitamins, cytokines, histamine, adenosine, energy, the molecules of the cell, the building blocks of the cells. These all interact, and it's all a continuous and very dynamic process. And there are unintended consequences of inappropriate immune activation. And so what I show here is I amplified that dendritic cell, that antigen presenting cell, and look at all of the things that it can be seeing at any given time with toxicities like aluminum salts. With the vaccinations that we're doing, and we're not thinking about those vaccinations as immune therapies or drugs or immune modulators, but what we had developed them as is mimicking infections. So what we had been doing was actually putting, and we still do it, is in a 2-month-old or a year-old child who doesn't have a fully developed immune system, and we'll talk about it, we're putting up to 3 different gram-negative bacteria. We're putting a few viruses like hepatitis B, and we're putting the actual either molecular subunit that is the antigen, along with an entire list of what we call adjuvants. Aluminum is one of them, and what that does is overstimulate the immune system. All of the bacteria have these nucleotides, CpGs, that are signaling molecules, and they're a sink for DNA methylation, for methylation, which is a key regulation pathway in all of these pathogens. So all of these things are interacting with the dendritic cell, which then talks to essentially everybody else in the innate immune response, and as I showed you in the last slide, the adaptive immune response. But we'll talk today about mast cells, and we've seen literature where antihistamines are actually helpful in treating, as part of a plan, Lyme disease and Borrelia infection. Well, why is that? Well, if this mast cell is inappropriately activated, then what you want to do is restore the balance of that cell so it's not producing all of this IL-9 and getting a subset of Th9 cells and ignoring the Th2 and the various other groups, Th17, that are supposed to clear the Borrelia. Because if your immune system's too busy doing it, can't do the rest of its job. And so we're actually crippling our immune systems. And this is a rather busy slide, but I wanted to get the information here for doctors in the audience to think about this. And what I say is advances in technology reveal unintended consequences of our current vaccine strategies. And I say vaccine, but there's also drug therapies and drug strategies, and we've just got to think about how the timing of how we apply these things and what the immune system of the person that you're giving them to looks like, because you can get quite unintended consequences. So in this 2014 edition of Trends in Immunology, they show this Matryoshka doll with the idea that the immune system is layered, that it's not at all static. It's not just looking like this, that an infant's immune system is highly immune suppressed, as is the mother. So what you see here is a gradient of TGF-beta, the most potent immune suppressant molecule in all cell types of the body. And so what you see is that the infant is immune suppressed up until about 2 years old, so that it tailors its immune response toward a Th2 response and can't mount any type 1 Th1 antiviral responses at all until it's between 2 and 3 years old. And we're going to talk a lot more because of the detox system, the kidney, the liver, they're not functioning fully. The gut microbiome is not functioning fully. And so what you get when you do the strategy that has only been developed in the last 20 years, where it seems that we can inject 8 different antigens full of aluminum and the various toxins that I showed you into a developing infant with no knowledge, or now with this knowledge, and that infant can't possibly develop an immune response, and all you can do is develop a dysregulated, what we would call an acquired immune deficiency. So out of the gate, it just makes, we'll hear the talk later today about common sense. And well, it's not really so common, because this is, when I tell my colleagues that this is what the vaccine industry does, because we're in cancer drug development or we're in AIDS drug development. I never developed a vaccine. And my colleagues will say, no, nobody do that. Why would anybody give a newborn hepatitis B? It's a pararetrovirus. That means it has a reverse transcriptase, so it can reverse transcribe your basic genome right out of the gate and set up an acquired immune deficiency. Why do we do that? It makes no sense. There's no way an infant can develop an immune response to these things until they're 3 years old. And those of us who are older, remember we were walking and talking in kindergarten when we got that one antigen at one time, and it was naked. It wasn't a needle full of a liquid full of toxins. So if you won't take a drink throughout your entire pregnancy, why would you inject a mother with these toxins? I mean, directly injected directly into the brain. So I show this this book you can buy this on Amazon. It's called The Immune System in the Developing Brain. A fabulous book to talk about the fact, it's a bunch of research that these editors from Duke University in the US put together the research to show that the brain system and the immune system are inextricably linked from the time pre-birth, that you cannot separate the developing brain and the developing immune system. So as you age, the immune system is changing. It gets better with age. This is what a 9-year-old's immune system is formed, all its circuitry, but it's not till it's an adult, and we all know what a teenager going through teenage years and puberty is like. So what we're doing now, and what this slide shows you, is that we're taking children in the most critical times of their development, and we're giving them this Gardasil, this human papillomavirus vaccine, And this looks like, you know, an infection. So you've got viruses, bacteria, you've got your innate immunity that I showed you, and these cytokine storms literally giving you the sickness syndrome of the fever, anorexia, fatigue, withdrawal. And what we found, and this is just, it's just a horrible paper. I could hardly read through the end of it without throwing it at my husband. He's used to it. But what they said, that this is a hypothesis paper, and they said, look, we're finding out that human papillomavirus vaccination is causing this new syndrome called HPV vaccination syndrome. It's a small fiber neuropathy and dysautonomia, and that could be its underlying pathogenesis. And the last line of the paper says, oh, we can find new biomarkers and new treatments for this new disease. And I'm like, just stop injecting a teenager. You know, where's the common sense? It's crazy. And what we've also learned is what we always used to say is that the brain and the eyes were immunologically privileged sites. And what that means is they can't tolerate, they couldn't tolerate the introduction of antigens without eliciting an immune response. So immune tolerance or immune privilege means that these sites, if you introduce these antigens downstream, you had a blood-brain barrier. If you injected the child or the teenager, it would not go directly to the brain. Well, that's simply, this research and the research in those two books in that chapter showed you that's simply not true. And then earlier research, so you actually do produce an inflammatory response. And when you get that inflammatory response, the entire energy of the organism goes to clearing it, especially in the infants, so that you because the infection and the inflammation will kill the host. We don't have, and it takes its attention away from development. You have to worry about developing if you die from an infection. So what else? And then you'll see the development go back. And so in diseases like autism and things like that, where these infections, and we'll hear a lot about it today that we're from Dr. McDonald, that Borrelia and these forms can go directly to the brain, and that's a real problem for Dr. Schwartzbach. So we have also identified through technology new primary immune deficiencies. And this was identified a year or so ago by Mike Leonardo, who was also an AIDS researcher. And what he shows, what he calls this is X-linked magnesium, EBV infection, and neoplasia. So there's a primary immune deficiency that's linked to the X chromosome, so women, and these are all the symptoms. Recognize any of them? Sinusitis, chronic EBV, splenomegaly, active herpes virus infections. And what they found out was the magnesium transporter, shown here called MAGT1, is supposed to activate a cascade that tells the natural killer cells to kill. And without this cascade and without this transporter, they don't kill. They're there and they don't know what to do. And so this is correctable in some cases simply by adding magnesium in the right form. So we'll hear a lot about that today from Dr. Valonen, as far as thinking about ions and the important balance of calcium, zinc, and magnesium ions in immune regulation, which we've only just learned about. Second discovery is the glymphatics or lymphatic system. It was always thought that the blood-brain barrier and the lymphatic stopped right here, but new technologies of imaging, since they were so close to the capillaries, the green indicates there the lymphatics. And it's not just a drainage system, it's actually a pumping system to cleanse the brain. So when you do an infection or an immunization, the immune cells surround the bacillus or the organism as they're supposed to do, and they traffic through the lymphatic system, and you can get vasculitis, inflammation of the vasculature, if that system's overwhelmed and you can't break down those, but they go directly to the brains. The toxin goes directly and out the sinuses. So it starts to put a connection to this, and we've done a lot of research in breast cancer and a lot of cancers where lymphedema is a real big problem. So we have homeopathic remedies that can actually cleanse the brain and cleanse that lymphatics and do a lot towards enabling some of the treatments to get to the brain. So Finally, what we've discovered, this is what we think about with HIV all these years, the war and peace between microbes. So what I say here is the Borrelia interactions with co-infecting viruses, which we'll hear from Armin next, vaccines, and the environmental toxins, which we just described. Well, what we've learned about the gut microbiome, the lung microbiome, the sinuses, the mouth, they're all different microbiomes. And what's really cool about it is different areas have different compartments. And when you break down that compartmentalization with toxins, with aluminum, mercury, other things in the environment, when you break that compartmentalization, the bacteria go everywhere, and they're not supposed to. They're supposed to be doing a job as a sentry right there. And so this changes such that as you age, you lose the equilibrium. Here's a nice little balance. You're at a balance with this symbiotic, our herpes viruses, other things that we've co-evolved with for years, and then something comes along, a virus, Borrelia, and it breaks down the balance. And so once it breaks down the balance, you no longer have, it's sterile inflammation at all. We used to call it sterile. Well, there's no sterile inflammation. It's all important in knowing about it in the treatment and prevention of chronic disease. So just to summarize, what we think about is a disease engine. And so the disease engine here starts with subclinical. There are epigenetic and genetic things going on, and we get damage from a distance, as I've talked about, things going to the brain, things going to the lymph nodes in the periphery, and that stimulates oxidative stress, the cytokine storm, and just a circle and a disease engine that if you don't stop the process somewhere in the middle, then you end up with disease. And it's all of these things on the side, the acquired immunological susceptibility I've just described, along with gut dysbiosis, this leaky gut. And this happens from early life, and it changes throughout late life, where everything has to be considered that goes to the gut, goes directly to the brain, as we know. So in conclusion then, to step outside the box, we're considering chronic neuroimmune diseases emerging in the last 4 decades as acquired immune deficiencies, because that's what they are. Because if you have a normal functioning immune system, you'll clear these, you'll silence these, and you'll be, or the antimicrobials will be able to help you do that. And it's those patients who can't get, even with months of treatment with things. And so what we look at is the families. Why is one person sick when the environment and some of the genetics and a lot of what is going on? And so we can learn a lot by studying the families, and we'll hear a lot about that today with the case reports. So we can use these advances in technology to drive these paradigm shifts and understand basic biology, and it affords us an incredible opportunity to end the suffering worldwide. And I just want to say, when I sat in a room in 1980 and I started seeing the AIDS patients come in, I couldn't imagine anything more horrific until I saw the children. And I never thought we would turn that into a quality of life disease that we could treat and you could live with, if not perfectly well, but much more well than almost everybody in the room. And we did it. So we can do it. We have the technology, we have the doctors, we have the motivation, and this is a good starting place to do it. Thank you.